Hey guys, so today we're going to be talking about the history of forensics. This is for forensics and its use in biotechnology as a part of our Biotech 1 study. The first thing that we're going to talk about is the history of fingerprinting. So, fingerprinting was the first application of forensics. The ancient Chinese used fingerprinting to identify different business documents. A lot of times you would have seen letters sealed with like wax, and they would have put a thumbprint in it or some sort of stamp or seal. This is kind of one of the first evolutions of fingerprinting. And then in 1892, a eugenicist named Sir Francis Galton established the first system for classifying fingerprints. In 1896, Sir Edward Henry, the Commissioner of the Metropolitan Police of London, developed his own system based on the direction, flow, pattern, and other characteristics. And Sir Edward Henry's system is one that we're still using today when we're analyzing fingerprints. You might be asking yourself, how do fingerprints transfer? They transfer from your hands to different surfaces based on the oils that are on your hands and on the porous nature of the surface that you're touching. And that's something that we'll talk about a little bit more in our activities as we go through the unit. There are three different types of fingerprints. The first one is arches. Arches occur in about 5% of fingerprints. Ridges of the fingers run continuously from one side of the finger to the other, making no backwards turns. There are subcategories of these arches. The first one is called a plain arch, and you can see that right down here in the bottom left-hand corner of your screen. A plain arch starts on one side of the finger, and the ridge slightly cascades upwards. Or you have a tinted arch. That's right here in the middle of the screen. A tinted arch lies in the ridges in the center and is not continuous like a plain arch. So you can see that these arches are a little bit disjointed. They're not continuous, but that they climb into the center and then kind of stop. And it's a little bit more peaked than the plain arch. You'll notice over here on the plain arches that a lot of these arches are continuously moving across the page. Take a look at your fingerprints. Can you see any arches on your hands? Are you one of the 5%? The next type of fingerprint is called a loop. This can be seen in about 60 to 70% of fingerprints. Ridges make backwards turns in loops, but they do not twist. Backwards turns are distinguished by how the loop flows on the hand and not by how the loop flows on the card when the imprint is taken. So we have three subcategories of loops. The first one is an ulnar loop. These loops are not very common, and most of the time will only be found on the index finger. And there's an example of an ulnar loop right down here on the right-hand bottom corner of your screen. There's also radial loops. The flow of this pattern runs from thumb towards the little finger of the hand, and we have an example of that on the bottom right hand of your screen as well. You'll notice that the ulnar and the radial loops are actually running in opposite directions, and that's one of the ways that you can tell them apart. And then we also have the subcategory of a double loop. A double loop is two distinct or separate loops, and where at least one recurring ridge within the inner pattern to form the two loop or the double loop pattern. And then over here in the bottom left, we just have a simple loop style kind of showing you what it looks like. The last of our three types of fingerprints is called a whorl. A whorl can be seen in about 25 to 35% of fingertips. Some of the ridges in a whorl make a turn through at least one circuit. There are three subcategories. The first is a plain whorl, where the ridges in these whorls make a turn of one complete circuit with two deltas and are therefore circular or spiral in shape, or spiral in shape, I'm sorry. And these are the simplest and most common of the whorls. There's also a central pocket loop whorl, which consists of one recurring ridge on an, or an obstruction at the right angle to the line of the flow. The whorl ridge may be oval, circular, spiral, or any variant of a circle. And then the last one is an accidental whorl. This is derived from two distinct shapes of patterns that have at least two deltas. Therefore, the whorls containing ridges that match characteristics of other subgroup whorls are accidental. Now, things that we want to talk about is how fingerprints are left behind. And I kind of alluded to that when we started, when we were talking about the oils on your hands. So there are different types of fingerprints that can be left behind. One is an imprint. This is a fingerprint that is left in a soft surface like wax, like the ancient Chinese used, or soap. Think of like bar soap, that kind of like softer, more malleable kind of surface. You also have patent fingerprints, which are visible to the naked eye. These would be things like your hands have dirt or ink on them and you're able to see your fingerprints through them. That would be a patent fingerprint. The one that we're most concerned with as forensic technologists would be the latent fingerprints. These are invisible. These invisible latent fingerprints are made of water, fatty acids, and amino acids, and triglycerides, or in other words, they result from the oils and sweat that your skin produces naturally. To make them visible, you have to find a way to detect one of these substances present in the invisible fingerprint. The easiest method is called dusting. And if you've ever had a police officer come out to investigate whether your house was broken into or your car was broken into, they may have dusted for fingerprints. And it's an incredibly fine dust material. It's kind of like a powder. And it sticks to the oils in the fingerprint. Once the fingerprint becomes visible, you can lift it from the surface using clear tape and transfer it to another surface to then take into the laboratory to analyze further. We're going to switch gears now and talk a little bit about ballistics. Ballistics is the study of projectiles in flight, which includes the launch and the behavior of the projectile. In 1835, Scotland Yard's Henry Goddard became the first person to use physical analysis to connect a bullet to a murder weapon. Bullet examination became more precise in the 1920s when American physician Calvin Goddard created the comparison microscope to help determine which bullets came from which shell casings. And then in 1970, a team of scientists at the Aerospace Corporation in California developed a method for detecting gunshot residue using a scanning electron microscope. So what we can see here is that with the development of several different types of microscopes, we're able to study and further analyze smaller and smaller evidence each time. So the comparison microscope is huge because it allows you to look at side-by-side pictures of a bullet that was fired that was collected from a crime scene and one that was fired into a water tank. And the markings on them should be completely identical if they were fired from the same weapon. The scanning electron microscope is really cool because it allows you to detect even the smallest changes in things due to those electrons, which is one of the smallest units of matter. So a little bit more about the comparison microscope and the scanning electron microscope. The comparison microscope is the one you see down here on the left side of your screen. The scanning electron microscope is this whole desk setup that they have over here on the right-hand side of your screen. A comparison microscope is a device used to analyze side-by-side specimen. It consists of two microscopes connected by an optical bridge, which results in a split view window enabling two separate objects to be viewed simultaneously. Where the scanning electron microscope is the type of electron microscope that produces images of a sample by scanning the surface with a focused beam of electrons. The electrons interact with atoms in the sample, producing various signals that contain information about the surface topography and composition of the sample. Did you know that all firearm barrels are unique? They have what's called grooves and lands. Grooves are spiral cuts in the rifled bore, and lands are the ridges of metal between the grooves in the rifle bore. And these are important because they make completely unique characteristics as the bullet is fired out of the weapon. These lands and grooves cause the bullet to spin in the air, and as the bullet travels through the barrel, a unique pattern is left on the bullet called rifling. Rifling is like a fingerprint and is completely unique to the firearm. There are 3 different types of ballistics. The first is internal ballistics. This is the study of the process that occurs inside of the firearm when a shot is fired. Basically, anything that happens from the time the trigger is pulled until the bullet comes out of the barrel would be considered internal ballistics. External ballistics is the study of the projectile's flight from the time it leaves the muzzle of the barrel until it hits the target. This determines things like trajectory of the projectile and the maximum range of a particular firearm. Lastly, there's terminal ballistics. This is the study of what happens when the projectile hits a target. It can help you determine the caliber, type of bullet, and distance between the shooter and the victim. It also can look at ricochet possibilities and damage caused from a specific weapon. In internal ballistics and how bullets are used in forensics, we use the lands and grooves to match the unique fingerprint or rifling using a comparison microscope to compare 2 bullets side by side. Firing pin marks can also be compared the same way as lands and grooves. Markings on cartridges can be utilized, and even serial number restoration are all part of the internal ballistics. In external ballistics, we're looking at how gunshot residue might be used. So when a firearm is fired, smoke particles from the gunpowder get all over nearby surfaces. GSR, or gunshot residue, can leave evidence that is very difficult to get rid of. It contains nitrates, lead, barium, and antimony, which are sticky particles that can be detected even after washing hands. Investigators will oftentimes test a suspect's hands to determine if GSR is present. To do this, they use a scanning electron microscope and X-ray spectrophotometry. Now, the portion of forensics that we're going to spend the most time discussing, because it most tightly relates to what we talk about in our class, is that of DNA testing. And the history of DNA testing goes back quite a long way to 1836, when a Scottish chemist named James Marsh developed a chemical test to detect arsenic. In the 1930s, scientists Carl Landensteiner won the Nobel Prize for classifying human blood into various groups. His work paved the way for future use of blood in criminal investigations. In the mid-1900s, other tests were developed to analyze saliva, semen, and other bodily fluids, as well as make blood tests more precise. In the early 20th century, law enforcement realized it needed a special team to analyze evidence found at crime scenes. Edmond Locard, a professor at the University of Loins, set up the first police crime laboratory in France in 1910. And that is where we really start to see forensics take shape. Auguste Vollmer, chief of the LA Police, established the first American police crime laboratory in 1924. Fun fact, is when the FBI was formed in 1908, it did not have its own forensic crime laboratory. That laboratory was not set up until 1932. So now that you know a little bit about how we started getting forensics labs and when we started analyzing blood and evidence that were left behind, we're going to go back and talk about the blood. So one of the first ways to distinguish between individuals was through ABO blood typing. This technique is incredibly fast, but has a low power of discrimination because it only divides people up into 4 subcategories, A, B, and O, as we have our 4 different blood types. So it's only able to separate people into very large groups, not individuals. The next technique that was developed was the RFLP, or restriction fragment length polymorphism. This is a very time-consuming process, but has a very high power of discrimination. It has the ability to identify a person on an individual level. Today's preferred method, however, is the STR analysis, or the short tandem repeat analysis by PCR. This is both fast and highly discriminatory. It uses variant spacer DNA, known as introns, to distinguish from one person to another. This is the method that we will be using through our labs for this unit as well. So you may want to highlight or write down that the method that we use is the short tandem repeat, or STR analysis by PCR. Restriction length polymorphism started out with restriction enzymes. We know from previous studies that restriction enzymes are a vital role player in biotechnology, and especially for studying DNA. Remember that restriction enzymes can cut DNA into manageable pieces for analysis. This can reveal information about the differences in DNA sequences. Restriction fragment length polymorphism in forensic application, this was the first DNA fingerprint used. And I don't mean fingerprint like the one we're getting off of your finger. Think of DNA fingerprint as a barcode that is use specific. So just like we go into the store and we scan a barcode to purchase our items, and that barcode is specific to the item that you've purchased, your DNA fingerprint is the genetic information, or the barcode, for your life and how your body or how your organism is genetically made up. A section of DNA that is known to differ in individuals is subjected to restriction digestion analysis, again through restriction enzymes like we learned about in previous units. When the restriction digestion is run on a gel, a unique banding pattern will form, or what we call a fingerprint, is seen. Only 2 individuals who might share the exact DNA fingerprint are identical twins, or asexually produced clones. So this is basically telling you that RFLP has an incredibly high power of discrimination. It can distinguish between any 2 people on Earth, with the exception of identical twins. When the PCR was developed in the 1980s, we started using it for DNA analysis. This started close to the 20th century, and we know that the PCR can make millions or billions of copies of DNA from a tiny sample of genetic material using markers. DNA is a unique genetic fingerprint that distinguishes one person from another. Remember that no 2 people have the same DNA except for identical twins. Today, forensic scientists can identify a person from just a few tiny blood drops or tissue cells. Other techniques that we're going to use in our forensic study will be spectrophotometry. This is used to identify unknown drugs that are seized, and infrared spectrometry, which is used to identify the structure and chemical components of soils, paints, and fibers that may be helpful in crime scene analysis. We might also use gas chromatography or mass spectrometry, which isolates the drug from a mixing agent or substance that might be mixed in it. Thank you guys so much. Make sure that you go back through and highlight any of your terms or any important information. Write down questions in the margin, and I would be happy to address them before our quiz tomorrow. See you guys later.